Hello friends, myself Dr. Nitin Khandare, Assistant Professor and Head Department of Botany, Sri Vasantrav Naik Mahavidyalaya Dharni, District Amravati. Today's topic is restriction enzymes. Steward, Linn and Werner Arber in 1963 isolated two enzymes which restricted the growth of bacteriophage in bacterium E. coli. One of these enzymes added methyl group to DNA and second one cut DNA. The second enzyme was named as restriction endonuclease. H.O.Smith and K.W. Wilcox and T.J. Kelley in 1968 isolated restriction endonucleases whose working dependent on a particular nucleotide sequence. They isolated this enzyme from bacteria Haemophilus influenzae and called it as Hind II. It was observed that Hind II always cut DNA molecule at specific place by identifying a particular sequence of 6 base pair. Restriction enzymes belong to a larger class of enzymes called nucleases. They are of two types: exonucleases, they remove nucleotide from ends, and second one endonucleases, they make cuts at specific position within the DNA. Thus, restriction enzymes or restriction endonuclease recognize a specific base pair sequence in a DNA called a restriction site and cleave the DNA, means hydrolyzes the phosphodiester backbones within the sequence. Restriction enzymes are widely found in prokaryotes and provide protection to host cell by destroying foreign DNA that make entry into it. Here, they act as a part of defense mechanism called Restriction Modification System. It bears two components. First component is a restriction enzyme that are selectively identifies a specific DNA sequence and degrades any DNA bearing that sequence. In second component is a modification enzyme. It adds a methyl group to one or two bases within the sequence identified restriction enzyme. If a base in a DNA is modified due to addition of methyl group, restriction enzymes cannot identify and cleave that DNA. By this method, bacteria are able to protect their chromosomal DNA from cleavage by restriction enzymes. Here, the diagram showing how these restriction enzymes cuts the DNA and ultimately the DNA between bases G and A only when sequence is present in the DNA. Thus, bacteria bears a set of restriction endonucleases and corresponding methylases. Endonucleases are enzymes that produce internal cuts called cleavage in DNA molecule. Endonucleases cleave DNA molecule at random sites. A class of endonucleases cleaves DNA only within or near those sites with specific base sequence called restriction endonucleases. Sites recognized by them are called recognition sites or recognition sequences. These sites differ for different restriction enzymes. Restriction endonucleases serve as the tool for cutting DNA molecule at predetermined sites, which is basic requirement for gene cloning or recombinant DNA technology. Here, the diagram showing the hydrolysis of the 3 phosphate bond in a nucleotide by a restriction endonuclease. Types of restriction endonuclease. There are 3 main types of restriction endonucleases, that is type 1, type 2 and type 3 are known with slightly different mode of action. Type 2 restriction enzymes are used in rDNA technology because they can be used in vitro to identify and cleave within specific DNA sequences, usually having 4 to 8 nucleotides. More than 350 different types 2 endonucleases with 100 different recognition sequences are known. They need magnesium dipositive ions for cleavage. The first type 2 enzymes isolated was Hind II in 1970. Here the table showing some examples of type 2 restriction enzymes along with source and recognized sites on sequences. The recognition sequences for type 2 restriction enzymes from palindrome with rotational symmetry. In a palindrome, base sequence of a second half in DNA strand represent the mirror image of base sequence of first half. Due to this, in DNA double helix, complementary strand also represent the same mirror image. Here the single strand, here the double strand. Palindromes are group of letters that form a same word when read both the forward and backward. Example Malayalam. As against a palindrome when same word is read in both the direction. Palindrome in a DNA is a sequence of base pairs that reads same of the two strands when orientation or reading is kept the same. However, palindrome with rotational symmetries, second half of complementary strand in a DNA double helix is the mirror image of a base sequence in a first half of another strand. In such cases, base sequences in both strands of DNA helix represent the same when read from same, and that is either 5' or 3' of both strands in DNA duplex. Example here, the echo R1 recognition site, an example of palindrome with rotational symmetry. Echo RI cleavage DNA molecule of two plasmid due to similar recognition sites in their DNA. The circular form of DNA becomes linear in both the cases. Such linear DNA can stick together to form single recombinant DNA molecule. Then the nomenclatures. Nomenclature of restriction enzymes is usually done by following techniques. The first letter of the genus is taken in which said enzymes was discovered. This letter is written in capital. Then first two letters of species of that organism are written. Then third, all the above three letters should be written in italics. Numbers of following the names indicate the order in which enzymes were isolated from strain of bacteria. If an organism forms many enzymes, they are identified sequential Roman numerals. Example echo K. Enzymes formed by H. influenzae strain RD have been named as Hind II and Hind III etc. Discovery of enzymes of EcoRI led to award of Nobel Prizes to W. Arber and H.O.Smith and D. Nathans in 1978. Types of cleavage produced by restriction enzymes. Many restriction enzymes like small isolated form Serratia mercanus have cleave both the strand of DNA at exactly same nucleotide position, almost in center of recognition site, resulting in a blunt or flush end. Small recognizes the 6 nucleotide palindromic sequence and cleave at the both ends. Still, some other restriction enzymes cleave the recognized sequence asymmetrically. Thus, due to the cleavage, they produce short single-stranded hanging structure. Such ends are called strictly or cohesive ends because base pairing between them can stick the DNA molecule again. A 6 nucleotides palindromic nucleotide sequence recognized by EcoRI cleave both the strand at different points. So, thank you for watching this video lecture. Please subscribe my YouTube channel for upcoming video lectures. Thank you.